Welcome back to another video on my channel. Sorry for not uploading, but I'm back with another walkthrough. Today we will be going through the AQA Combined Science Trilogy paper, and it's the higher tier Biology paper 1. Okay, so question 1. A student investigated the effect of different concentrations of sugar solution on pieces of potato. This is the method used. Cut 5 pieces of potato, record the starting mass of each piece of potato, place each piece of potato in a different concentration of sugar solution. After 24 hours, remove the pieces of potato from the solutions. Record the final mass of each piece of potato. Calculate the change in mass for each piece of potato. The question is, what is the independent variable? It's really important that you underline command words, or you can box command words and highlight key information, key pieces of information that you see in the question. Tick one box. So just taking you through the variables, independent variable is the variable I change in an investigation. So the way to remember is independent, letter I, what I change in an experiment. So what has this student changed during this investigation? So change in mass, concentration of sugar, length of time, starting mass of the pieces of potato. So if we go back to step 3, different concentrations of sugar solution. So we're changing the concentration of the sugar solution. Table 1 shows the results. So you've got concentration of sugar solution in moles per decimeter cubed, mass of potato at the start, mass of potato after 24 hours, change in mass in grams. Explain why the potato in 0.0 moles per decimeter cubed sugar solution increased in mass. So if we just highlight that row, so we know that we're looking at that row. So explain is the command word. Why the potato increased in mass. So 7.94 at the beginning, 10.14 at the end. So the reason why there was an increase in mass is because the potato gained water. Okay, and I've said this before in my previous walkthrough that it's okay to write in bullet points. Okay, just stick to the point. No need to write in full sentences. No need to copy out part of the question. Just go straight in and put your answer down. Now, this is an explained question, which means that you have to have science behind your answer in terms of, okay, so we're saying gained water. Why did it gain water? So it gained water by a process which we call osmosis. So we just say by osmosis. And that's it. 4 words gets you the 2 marks. Okay, so it's really important. Always have a look at how many marks are awarded for each question, and then according to the number of marks, you would have the same number of bullet points. Okay, right, next question. So complete figure 1. Some of the results have been plotted for you. You should plot the data from table 1. Draw a line of best fit through all the points. Now, it's really important that you pay attention to the scaffolding that they've provided, okay? And as soon as you've covered each point, just tick it off, because I've seen lots of students, when I mark their mock exams, that they do part of the question because they haven't gone back and looked at what the question required of them. Okay, so let's start plotting the results. So 0 is, so we're looking at change in mass in grams. So again, I find this useful. Just highlight the numbers that you're working with. 3 plots are already present. So 0 is 2.2. So 2.2 is there. Okay, the reason why I'm doing it in pen is so that you can see the plot, okay? But I would suggest when you're doing your exam, make sure the cross is in pencil. Okay, so 0.1 is 1.15, so that's 1.10, 1.20, so that would be in the middle there. And make sure your cross is the same size as this small square. Don't make your cross too big. And also don't use dots to plot, because when you draw your line of best fit, the dots won't be visible because they'll be underneath the line, so the examiners cannot see where your plots are. 0.2 is 0.25, so that's 0.1, that's 0.2, that's 0.3, so that will be there. And these are the 3 plots. Okay, so go back and just tick that off, right? I've done that. Draw a line of best fit through all the points. So, now if you have a look, okay, a good technique is to, obviously you won't be able to see this, but what you do is bring the paper close towards eye level and look at it at an angle, and you should be able to see that it's a smooth curve. So a line of best fit can be a line or it can be a curve. If it's a curve, do not use a ruler. If it's a line, use a ruler. If it's a straight line. Okay, so let's draw the curve. Don't let go. Keep going. And it should look like that. Okay, go back. Tick that off. Okay, and that should get you your 2 marks. Right, next question. The mass of a piece of potato does not change when concentration of solution inside the cells equals concentration of solution outside cells. Determine the concentration of sugar solution inside the potato cell. Okay. So determine is the command word. That's what the question is asking us to do. Okay, so the way we do this is, if you want to work out the concentration of the solution inside of a potato, or if this question was about a carrot, or if this question was about a boiled egg, the technique is exactly the same. So what you do is you look at where your line of best fit meets the x-axis. Okay, and you can see it's this point here. This is the value you need to read off. So if we have a look, okay, it's so 0.21, 0.22, 0.23 exactly. So we're looking at exact point at which the line meets the x-axis. So the answer is 0.23. Okay. Okay, so now we've got the same table again. So calculate the percentage change in mass for the potato in 0.2 moles per decimeter cubed sugar solution. Okay, so they've given you the equation, and it's advisable to go through, use a highlighter and just highlight the row that you're interested in so that you don't take the incorrect figures from a different row. Okay, so command word, they're asking us to calculate. So all you need to do is go to that row and take the relevant values. So change in mass is 0.25. There it is. Okay, we're dividing that by mass of potato at the start. That's 7.96. Multiply by 100. And if you put those numbers into a calculator, so 0.25, divide that by 7.96 times 100, we get 3.1407035. Write the whole answer down. Okay, right, now be careful here, you get an extra, so this is 2 marks. Okay, now the next thing you need to do is your answer should be written as 3 significant figures. So 3.14. Okay, and that should get you your 3 marks. Okay, now we're moving on to question 2. If I just take you back here again. Right, so question 1 was one of the required practicals. Okay, so that was the osmosis required practical. So we're looking at water movement in potato cylinders. That's the required practical you probably studied in your lessons, but do bear in mind that sometimes in exams it might not be a potato, it could be a carrot, okay, or it could be another vegetable, but the concept is the same. You just need to apply that knowledge to a different vegetable or a different fruit. Okay, as we're on the subject of required practicals, in biology paper 1 you have 5 required practicals. So if I just go through that quickly, so you've got microscopes. Okay, then you've got food tests, you've got enzymes and amylase. That was the amylase and starch one. Then we've got osmosis, and then the final one is photosynthesis. So remember, 15% of the paper has to be based around these required practicals. Okay, so please do have a look at that. Right, starch. So question 2, starch is digested to form sugar molecules in the digestive system. Okay, so starch is a long polymer. These circles represent glucose molecules, okay, and the whole thing is starch. So what is the name of the enzyme that digests starch? It's amylase. If you say carbohydrates, that is still correct. You should still get a mark for that. Question 2.2, where are most food molecules absorbed? Tick 1 box. Large intestine, liver, small intestine, stomach. It's the small intestines. Figure 2 shows 2 villi. Figure 2 also shows 1 cell on the surface of a villus as seen using an electron microscope. Okay, so you've got 2 villi, and then we've zoomed into one of the cells on the surface of the villus. And you can see you've got microvilli, there's mitochondria, and obviously you've got a nucleus there. Always use the diagram. There is a reason why there is a diagram there. Okay, give one advantage of using an electron microscope compared with using a light microscope. So have a think. Okay, so this image has been taken using an electron microscope. So like I said earlier on, look at the diagram and you should notice that this greater magnification. You won't be able to see this with a light microscope. So you could say greater magnification. You can also say, because if you look at that diagram, you can see subcellular structures, or you can see subcellular parts, because you can see mitochondria. You can't see that with a light microscope. You can also say you can see more detail. Okay, and then the final thing you could also possibly say is there is a higher resolving power. And again, and I've also said this previously in my previous video, that AQA, higher tier paper, even with foundation, they tend to give you lots of lines, and students think, gosh, I need to write a lot in order to get the 1 mark. You don't need to. You can see here, 2 words gets you 1 mark, and I haven't gone on to my second line. So just bear that in mind. You don't need to write a lot. What type of blood vessel is labelled X? So if we go back, so this is a blood vessel that lies very close to every single cell in your body. Okay, and substances need to be able to move in and out of this capillary. I was supposed to say in and out of the blood vessel, so I've given the answer away. It's capillary. The real length of 1 villus is 0.8 millimeters. Calculate, so box the command word, calculate the image length if the villus is viewed at a magnification of times 20. So we're looking at the image length, so we're working out this. Okay, so go in, substitute your values in. So magnification is 20. Size of image, I don't know. Divide that by 0.8. Rearrange 20 times 0.8 should get you the image size. So you put that into your calculator. That should give you 16. Okay, the units are already there. Bear in mind that this equation you should really memorize. It's not always given. Now this paper is 2020, so this was a while ago. This was the year we had COVID, so no one actually sat this paper, okay? But in future questions I have seen that this equation isn't always given, so I expect you to memorize that. Right, so that gets you 3 marks. Continuing on with the same question, figure 3 shows 2 cells from the surface of a villus. There are sugar molecules inside and next to each cell. So these are your sugar molecules, okay? You've got the key there. Name the process by which sugar moves into cell A. Okay, so before we continue, there are 3 processes that allow substances to move in and out. So diffusion, active transport, and then you've got osmosis. So let's start off with osmosis. Osmosis is basically diffusion, but it is the movement of water molecules only. Diffusion is the movement of any substance, okay, from an area of a high concentration to an area of low concentration. Always make sure when you're writing the definition, you use the word area. Now, active transport is where substances move from an area of low to an area of high concentration. And active transport requires energy because we're going against the concentration gradient, because normally it would be high to low. Where does this energy come from? It comes from, this energy is released via respiration, which occurs in mitochondria. So any cell in your body that carries out active transport, you will notice that those cells have a lot of mitochondria. Okay? So name the process by which sugar moves into cell A. Into cell A. So if you have a look, I've got way more sugar molecules on the outside, high, and I've got low on the inside. So the answer here is diffusion. Name the process by which sugar moves into cell B. So again, into. You can see there's low on the outside, high on the inside. So that must be active transport. Okay, 2.8. Give 1 use of sugar inside the body. Okay, so all of you should know this. The common use of sugar is respiration. So sugar combines with oxygen to release energy, produce carbon dioxide, produce water. There are other possible answers. You can also say sugar is converted into glycogen. You can say sugar is used to make amino acids or proteins, or it's used to make fat. Okay, but all of you should know respiration. Okay, the same diagram is repeated again. Explain how villi are adapted for efficient absorption of sugar molecules. So this is an explain question, which means that I would like you to write down the feature. So what is this special feature or this special adaptation? And then tell me the reason. What does that lead to? That's the explain bit of your answer. So if you look at the diagram, remember I said this earlier, the answer is there. There is a reason why they've given you the diagram. Use it. So let's start off with microvilli. So if that's one cell, then we know there's lots of microvilli throughout the small intestine. So bullet points have microvilli. Okay. Then tell me, yep, so what does this lead to? To increase the surface area. Okay, that's 2 bullet points done. So this is an extended writing question, and it's not, you've got 3 or 4 bullet points and you're probably going to get 4 marks. It's not necessarily marked like that. So the examiner will have a look, okay, and they will award either a level 1 or a level 2. So level 1 is where you've just basically just probably stated the features, the adaptations, but you haven't given the explanation behind it. So the maximum there would be 2 marks. But if you have given the adaptation and then you've given the reasoning behind that, what does that lead to? This is your explain bit, okay? And you've got 2 or 3 adaptations with the explanation, you would hit the 4 marks. Okay, so again, if you look at that diagram again, I can see there's lots of mitochondria. If that's one cell, can you imagine? There'll be so much more mitochondria throughout. So your next point could be that the cells have many mitochondria. Why? I've said this to you earlier on. This is where respiration takes place. It's really important. I know it's can happen sometimes, but just be careful on your actual exams. Do not go past the margin, okay? So this is where respiration takes place. So if you have a look at the diagram, what else can we see? Well, we've got X. Remember we labeled that earlier on, and that is a capillary. So we can say also there is a good blood supply. And so what? You could say this blood is continuously moving. Okay, so that maintains a diffusion gradient. So that is to maintain a diffusion gradient, which means that if the blood keeps moving away, it's bringing more sugar molecules in. So in the blood, we will always have high concentration of sugar, and then in the cells, it will always be low. So that sugar molecules will then move into the cell. So that is maintaining a diffusion gradient. And that should be enough. So if I just go through with my highlighter, and if I just, you don't have to do this, it's just me pointing it out to you. So the adaptations I've spoken about, microvilli, mitochondria, and a good blood supply. And everything else is the explanation bit. Okay? Just be careful, again, I said this earlier on, for level 2, which is 3 or 4 marks, you must make links between structure and the function. Structure, function, structure, function, structure, function. Okay? Say you just had this bit here. That should still get you 4 marks. I just wrote down 1 extra for you. Okay? Okay, so question 3. So being overweight can affect the health and life expectancy of a person. Give one disease related to being overweight. And there's quite a lot you can write here. Okay, I'm going to go for diabetes. Now be careful. If you write type 1 diabetes, that's incorrect. Okay, because type 1 occurs in childhood. This is looking at someone who's overweight. That leads to type 2 diabetes. Okay, so if you said type 2 diabetes, that would be marked correct. So just be careful, you know your ones and your twos, which you've studied in paper 2 as well. Other answers you can say coronary heart disease or just heart disease, high blood pressure, cancer, depression. You can even say arthritis. Right, 3.2, body mass index helps to show if a person has a healthy body mass for their height. BMI is calculated using this equation. You don't need to memorize this equation. If you have to calculate the BMI, this equation has to be given to you. Okay, a woman has a BMI of 27 and a body mass of 68.1. I would underline key figures. Calculate the woman's height in meters. Okay, so my command word is calculate. Okay, so the first thing, always show you're working out. Okay, because if you get your final answer wrong, you can always pick up marks for working out. So BMI is 27. My body mass in kilograms is 68.1. I'm working out the height. Be careful, height is squared, so put the squared there. Okay, and then I am going to rearrange. So 27 times h squared gives me 68.1. And to get h squared, that will be 68.1. Divide that by 27. Okay, so take a calculator. Okay, so there you can see that. So 68.1, divide that by 27. That gives me 2.522 reoccurring. Okay, keep that in your calculator. Square root that. And that gives an answer of, it doesn't say significant figures or anything, but I'm just going to go for 1.59. Okay, and that's meters. The unit is already there. So that's a nice easy question where you don't have to memorize the equation, you just insert your values. Okay, figure 4 shows a height body mass chart for adults. Which weight category describes a woman in question 3.2? Okay, so I just need her body mass again, so that was 68.1. So that's that axis, and her height was 1.59. Okay, so what you do is find 68.1, okay, which is roughly there. Keep going until you get to 1.59. Can't see that. And 1.59 probably there somewhere. So you go until where the two lines meet, and it meets just there. Yeah, and you can see it's overweight category. So you tick over weight. Okay, so that was another straightforward question. Now, if your height didn't come to 1.59 and you had some other random figure somehow, say you got, I don't know, 1.2, and you've used 1.2, but you've used that figure, but you've correctly identified the category. So maybe 1.2 is not even on there, but let's just say you got, I don't know, 1.8. Yeah, if we go up to 1.8, the individual is healthy weight, and if you ticked healthy weight, you will still get a mark for this. It's error carried forward. Okay. 3.4. People are encouraged to control their body mass with diet and exercise. Describe how the balance, that's your command word, describe how the balance between the mass of food eaten and the amount of exercise a person does controls body mass. Okay, so the food that we eat is basically energy intake. Okay, so we can say increased energy intake if the person eats more food. And then let's talk about exercise, and if they exercise more energy is transferred. So obviously more energy is being released. Okay, so that should get me 2 marks or 2 bullet points. And the third thing you could say is if energy intake equals energy transferred, body mass would stay the same. Okay, so increased energy intake if more food is eaten. If you exercise more energy is transferred. And if energy intake and energy transferred is equal, body mass should stay the same. Okay, or the other thing you can say here is if more energy is transferred than is taken in, body mass should decrease. Or the converse of that, if less energy is transferred than is taken in, body mass would increase. Okay. Okay, so 3.5. So this question is looking at the very last topic of biology paper 1, bioenergetics, and its respiration. So during long periods of vigorous exercise, the body respires anaerobically. Anaerobically is respiration, N, where there is no oxygen being utilized. Explain the changes that happen in the body during and after vigorous exercise. So for a 6 marker, you already know that we write in bullet points. You can also have subheadings. So we need to look at during and after exercise. So let's have a heading, during exercise. So we will write the change that happens with a reasoning. So it's an explained question. So, okay, so what's happening during exercise? We can say there's increased breathing rate. The next bit is why? To take in more oxygen for respiration. Okay, what happens? Heart rate increases, so we can say increased heart rate. Okay, and then now you need to tell me the reason for the increased heart rate. That is to transport oxygen and glucose to respiring cells, to transport oxygen and glucose to respiring cells. And I think that's enough for during exercise. As long as you've got a balance, like 3-ish bullet points for during, 3-ish for after exercise. So after exercise. Okay, so this is exercise is done. What's going to happen to the body? Well, the heart rate will still remain high. So heart rate remains high. Or if you wanted to, you can say the heart rate slowly decreases. So the reason for this is so that the body, the person continues to breathe rapidly to pay back oxygen debt. Okay, so heart rate remains high. Breathe rapidly. Okay, I believe that. Yeah, that's it. So continue to breathe rapidly. We can say also, if you wanted to, you can define oxygen debt, but we leave that. So breathe rapidly to pay back oxygen debt. And also, remember, during anaerobic respiration, there's build-up of lactic acid, which has to be removed. So we're breathing rapidly, heart rate remains high. This is so that there is more oxygen. So to provide more oxygen to react with lactic acid and remove it from cells. So remember, lactic acid is acidic, it lowers the pH of the blood, it's a poisonous substance which has to be removed immediately. So lactic acid reacts with this oxygen that we're taking in, which converts the lactic acid into carbon dioxide and water, which we obviously exhale. But this is enough. So I've got 4 bullet points there, 4 bullet points there, and I've got the full coverage there. Okay, so this is during and after exercise. So to get 5 or 6 marks here, so because it's a 6 marker, there's 3 levels: level 1, level 2, level 3. To get your level 3, which is 5 or 6 marks, you must talk about what happens to the body during exercise and after exercise, and you must mention lactic acid and its removal, which we have. Okay, basics, question 4 now. So we're going to the very first topic of biology, paper 1, which is cell biology. Cells are the basic units of all forms of life. Describe 4 differences between a bacterial cell and a plant cell. Okay, so this is a nice, straightforward question. So let's go with the obvious. So bacterial cells do not have a nucleus, yeah? Okay, or you can say bacterial cells don't have DNA inside a nucleus, or you can just say that. I always say to students, write down the answer, which requires very little writing, and you still get the 1 mark. Okay, bacterial cells have a plasmid. Bacterial cells do not have mitochondria. So remember, bacterial cells, there's no nucleus, there's no membrane-bound organelles. Okay, so no mitochondria, no nucleus, and no chloroplasts, which we can mention. You can also say bacterial cells do not have a vacuum. So the only organelle that you would see in common between both is ribosomes. Okay, so if you want to write about ribosomes, the only thing you can mention is that bacterial cells have smaller ribosomes, whereas plant cells have a larger ribosome. Okay, so that's 4 points, 4 marks. So gonorrhea is a bacterial disease. A new vaccine is being developed against gonorrhea. Describe how a vaccine would work to prevent gonorrhea. So this question is basically looking at how a vaccine works, and they've linked it to gonorrhea. Now, this question could mention any disease here. It could say measles, it could say COVID, it could say cholera, but your answer would be identical. Okay, so don't be thrown off when you get a question like this. It's just a question that's looking at the basics of how a vaccine works. Okay, so gonorrhea, it already says it's a bacterial disease. So the first step of how a vaccine works is you inject the dead bacteria or the inactive bacteria. Okay, so inactive bacteria, because the question already tells us it's a bacterial disease, inactive bacteria is injected. Keep it nice and simple. What happens, or what triggers the immune system, is the white blood cells are stimulated to produce antibodies. Okay, so that's step 2. Step 3, you could then say that the individual gets gonorrhea, again, you can say that the antibodies are made very quickly. So on re-exposure, antibodies are made very quickly. You can also mention, because that any reference to memory cells would also get you a mark. So you can say memory cells are made or they remain in the bloodstream. And then your fourth mark should be the bacteria is killed. Because a bacteria would be killed straight away on re-exposure. Now this, I would say, use this answer for any question that you get in terms of how a vaccine works. Now, if this question was referring to measles, you would just change the bacteria to virus, because measles is caused by a virus and not bacteria. But everything else would stay the same, and you would just, yep, that's it here. Virus is destroyed, because virus is not living, so you can't say killed. Viruses are destroyed. But most of the answer would stay the same. Okay, right, 4.3. Another disease is caused by bacteria. Another disease caused by bacteria, Salmonella food poisoning. In the UK, chickens are vaccinated against Salmonella to reduce the number of cases of food poisoning in humans. Explain how vaccinating chickens reduces the number of cases of Salmonella food poisoning. Okay, so again, this time, whatever you say, there has to be scientific reasoning behind it. So here you would say fewer bacteria in chicken, or you can say in the eggs. So fewer bacteria ingested. That's it. That should get you 2 marks. Okay. 4.4, give one way that the spread of Salmonella food poisoning from one human to another is controlled. Do not refer to vaccination in your answer. Okay, so how can we reduce the spread? You can say wash hands before preparing the food, but not just wash hands on its own, because that's not going to get you marks. So wash hands before preparing food. Or you can say wash hands after using the toilet. You can also say clean areas where a person has been ill, or do not shake hands with someone who has food poisoning. Okay, but we'll go with what the easiest thing is to write. The number of cases of Salmonella food poisoning is usually higher in the summer than in the winter. Suggest one reason why. So warmer weather, so bacteria reproduce faster. Remember, bacteria will reproduce rapidly in warmer conditions. So warmer weather, so bacteria reproduce faster. This question is about photosynthesis and food production. How can oxygen production be used to show the rate of photosynthesis? Okay. So how can oxygen production, well, oxygen is produced during photosynthesis, so how can we use it to show if photosynthesis is happening fast or slow? So we can say measure the volume of oxygen produced in a given time. Or you can say produced in 1 minute, or produced in 2 minutes, or produced in 30 seconds, as long as you've given a time frame. Okay, so produced in a given time. So that should get you 1 mark. Scientists investigated factors affecting the rate of photosynthesis in tomato plants. The tomato plants were growing in a commercial greenhouse in the UK during winter. Figure 5 shows the results. Okay, so we've got a graph here. I've got light intensity here, rate of oxygen production in arbitrary units. Basically, this is your rate of photosynthesis. The percentage of carbon dioxide in the Earth's atmosphere is 0.04%. Name the factor that is limiting the rate of photosynthesis at point X. Okay, so you should have studied the 3 graphs which looks at the 3 limiting factors, so light intensity, concentration of carbon dioxide, and temperature. Okay, so light intensity graph looks like that, concentration of carbon dioxide looks like that, and the temperature one looks like that. Okay, on your x-axis you have the actual factor, so temperature, carbon dioxide concentration, light intensity, and then on the y-axis is basically your rate of photosynthesis. So the straight line that you have for this graph, that graph, this graph here, and these graphs, the reason why it's a straight line, it's because rate of photosynthesis is pretty fast, okay? Because I've got plenty of light there. So from 0 onwards, the limiting factor will always be the factor that's written on the x-axis. Okay, so at x, it's the light intensity that is limiting photosynthesis, because as I increase it, rate of photosynthesis is increasing until I reach this point where I've got plenty of light, but hardly any photosynthesis. That means light is not the limiting factor now. Light is not stopping photosynthesis. It's not limiting photosynthesis. It must be something else. And that could be temperature, or it could be carbon dioxide. So the answer here is light intensity. Farmers growing tomatoes commercially try to control the rate of photosynthesis and make maximum profit. A farmer can control the temperature and CO2 concentration in a greenhouse. What is the minimum light intensity a farmer should use to get a maximum rate of photosynthesis? So, maximum rate of photosynthesis. So what is the minimum light intensity a farmer should use? So if we have a look, there's our maximum rate of photosynthesis, yeah? So here, just put it up. So that's my maximum. So let's bring that down and look at what light intensity that would be. So there, 20,000. 20,000 lux. Lux is the unit of light intensity. The light intensity you gave in question 5.3 may not give the farmer maximum profit. Explain why. So this is where we can talk about cost. So there is a cost for heating the greenhouse. Okay, so obviously that would lead to the farmer not making maximum profit. You could also say there is a cost for lighting, and then additional costs might exceed profit. Okay, so you have to refer to cost because you're looking at maximum profit. Explain the results. Explain the results when the light intensity was 0 lux. So we're looking at 0, this point here. So no light, no photosynthesis. So no oxygen is produced. So I already got 2 marks there. But remember, plants are also respiring. Okay, so oxygen is being used in that sense. So no oxygen is produced via photosynthesis. Respiration is still occurring. Respiration happens all the time. and oxygen is used. And then we need to talk about why the line is in the negative axis. So the overall, or the net, so the overall oxygen production is negative, or you can read the value of it's -2. Okay, so I've got 4 bullet points there. So this part of the question is challenging. Okay, so new drugs are tested and trialed before they can be licensed to treat patients. So this question is looking at infection and response, and the topic is drug testing. Figure 6 shows how many times the different stages of testing took for one new drug. So you've got preclinical testing, clinical trials. So preclinical testing, this is where they are testing on animals, cells, and then they have computer models. Clinical trials, this is where human volunteers come in. So clinical trials start off with healthy volunteers, and then towards phase 3 they start looking at volunteers or patients who have that particular the particular disease where the medicine works for. Okay, review and approve. So how much more time did the clinical trials take compared with the pre-clinical testing? Right, so for this you need to, let's have a look at where shall we start? Right, let's have a look at preclinical testing. So preclinical testing, that takes 3.5 years. Yeah, this one here, 1 2 3, that's 4 years, that's 1.5, and that's 1. Okay, so let's add up the clinical trials. So that's 4 plus 1.5 plus 1, that gives me 6.5 years. Okay, and then preclinical testing is 3.5 years. The question is asking me how much more time did the clinical trials take. So 6.5 minus 3.5 gives me 3 years. So 3 years is the answer. During phase 1 clinical trials, low doses of the drug are tested on healthy volunteers. Suggest one reason why low doses of the drug are tested in phase 1 clinical trials. So here we can say, it's only 1 mark, to reduce any risk or to look for side effects. So like I said earlier, you can say to reduce any risk. Any idea that less harm will be caused with a lower dose. 6.3 suggests 2 reasons why healthy volunteers are used in phase 1 clinical trials. So here, so it's 2 marks, you can say too great a risk for ill person or ill patient. Okay, or you can say may make their condition worse, or you can say less risk to healthy person. And the second reason you could say is the patient, can't spell, patient might be taking other drugs, so you don't want the two drugs to react. I do apologize, not sure what's wrong with my spelling. Patient might be taking other drugs. 6.4, the results of clinical trials can only be published after peer review by other scientists. Suggest one reason why the results may be reviewed by other scientists. So that is to prevent false claims, or you can say to avoid bias. Okay, it's just 1 mark. So whenever you get a question like this, it's always about preventing false claims, or you can say to avoid bias. Okay, so on our last question here, so 6.5, a drug is only licensed for the medical conditions it was tested to treat in the clinical trials. Drug regulations control what drugs a doctor can prescribe, ensure doctors can prescribe a drug with confidence, protects patients. AMD is an eye condition that can result in very poor vision. Doctors treat approximately 40,000 new cases of AMD each year. Two drugs licensed to treat AMD in the UK are drug A and B. In many other countries, drug C is used to treat AMD. Drug C is only licensed in the UK to treat cancer. The cost per injection for each drug is as follows. The number of injections required to treat AMD is the same for each drug. So that's a lot of information you have to digest. In 2018, the High Court in the UK gave permissions for drug C to be used to treat AMD. Evaluate the decision to allow the use of drug C to treat AMD in the UK. Now, evaluate question, you need to have arguments for, arguments against, and you have to have a justified conclusion at the end. And there's no wrong or right conclusion. It's, what do you think? So let's go for arguments for. Again, you can have subheadings. And again, if you go back and look at that information again, so use that information, we can see that the for is it will save NHS money. I would say just have a balance, maybe 3 or 4 bullet points for each. So I'm just going to go for 3 bullet points. Okay, will save NHS money. You can say more people treated for the same cost. You can also say patients would be treated sooner. And other things that you could say is you can say more people can be treated for the same cost. I've already got that. Likely to have been tested in other countries, used in other countries, so it's likely to be safe. Okay, and then arguments for against. The injections of drug have not been tested in the UK. So cannot be sure if it's safe to use. What else could you say? You can say goes against regulations because all drugs must be tested. Okay. And then you can say your final conclusion. So I conclude, okay, like I said, there's no wrong or right answer here. So I conclude that it is a good idea to use drug C. To use drug C because, and what you do is you just pick some of the points and just rewrite them out again because it will save NHS money. So you're just backing it up with some of the points that you already made previously. Okay, and then that again, because it's a 6 marker, it's the examiner would award one of the following levels: level 1, level 2, level 3. And if your answer looks like this, it's definitely level 3 because I've got the for, I've got the against, and I've got a conclusion. And again, the conclusion can be, there's no wrong or right answer for this. So you might have said, I conclude the using drug C is not a good idea because it goes against drug regulations. Okay, so that's just specific to you. Okay, so that brings us to the end of the biology paper 1, and this was the May 2020 paper. What I will do is I will also upload the blank version of this paper plus the mark scheme, so you can have a go at the paper at your own time. So thank you for listening. Hopefully I will be back with another walkthrough, and I'm hoping to go through another physics paper, and thank you for all the comments that you've been posting too. I will come back with shorter video clips just going over specific areas that you want me to go over, so such as radioactive decay and specific latent heat. So thank you very much. Thank you for listening, and please do leave comments and any suggestions or any particular paper you want me to go through, any particular topics, and I will work on those areas. So thank you so much.